My name is Peter Armenta, and I have the honor of serving as Dean of this Rutgers Robert Wood Johnson Medical School. And as I've been saying that a lot this year, this is the first Black lecture, I believe, Ira Black lecture that is under Rutgers' banner now. So it's a wonderful day. I want to welcome you to the third annual lecture in memory of Dr. Black. Third. This is not third. It is third. I thought it was more than that. Ira, of course, was professor and chair of the Department of Neuroscience and Cell Biology at Rutgers. He was a world-renowned leader in the field of neuroscience and our founding director of the Stem Cell Institute of New Jersey, as well as the past president of the Society of Neuroscience in North America. Ira, as we know, passed away sadly and unexpectedly in 2006. And I know so many of you in the room knew and respected and loved Ira, and we still miss him till today. Over the course of his lifetime, Ira's research focused on molecular mechanisms that underlie brain function and cognition. He was a pioneer in the study of development of the peripheral nervous system, as well as the role of growth factors in that system and the brain. He also focused his attention on the potential of stem cells in brain repair. His team succeeded in converting adult bone marrow stem cells into nerve cells in culture and in vivo. And I have this vivid memory of coming home one night, and the TV was on, and Ira was on the national news talking about his stem cell work. His hope was that his work would increase understanding of the mechanisms that assist brain recovery from trauma and degenerative and acute neurological diseases, including Alzheimer's, Parkinson's, and spinal injury. Dr. Black is also remembered as a dedicated teacher and mentor, attributes which attracted some of the nation's best and brightest graduate students and young faculty members to our school. I'm pleased to welcome Janet Black, Reed, and Jenny Black back to our campus again this year. I'd like to express my appreciation to Janet for endowing the lectureship. I also want to thank Dr. Joseph Coyle, the Eben S. Draper Professor of Psychiatry and Neuroscience at Harvard, especially for today. I'm sure you had a lot of fun getting here. I look forward to hearing his presentation today. And now it's my absolute pleasure to turn over the program to Dr. Cheryl Dreyfus, our chair of the Department of Neuroscience and Cell Biology. Cheryl?
We were just thinking about Ira today and remembering that it was actually 8 years ago that many of us had our last conversations with him. It doesn't feel like it can possibly be so long ago, and that so much has happened since his death, to us and to our school. Ira was an excellent scientist and teacher. Moreover, he changed the neuroscience landscape on our Rutgers and Robert Wood Johnson campus by hiring a host of excellent scientists and raising awareness of neuroscience as a field to be explored. This mission is one that we continue to embrace. It's especially appropriate then that today, one of Ira's dear friends, Dr. Joseph Coyle, is here to speak to us and present the Ira Black Memorial Lecture. We welcome the Black family, and we thank you, Janet, for making this lecture a reality. Joseph Coyle holds the Eben S. Draper Chair of Psychiatry and Neuroscience at Harvard Medical School. From 1991 to 2001, he served as chairman of the Consolidated Department of Psychiatry at Harvard Medical School, which included the 9 hospital programs of psychiatry affiliated with Harvard. He has won numerous awards, including the Leiber Prize for Schizophrenia Research, and most recently, the Julius Axelrod Award that honors a scientist with distinguished achievements in the broad field of neuropharmacology, and so importantly, exemplary efforts in mentoring young scientists. He is the past president of the Society for Neuroscience and past president of the American College of Neuropsychopharmacology. He serves on the board of scientific counselors of the National Institute of Mental Health. He serves on more than 20 editorial boards, including JAMA, and is the editor-in-chief of JAMA Psychiatry, the most highly cited journal in the field. His research interests are in developmental neurobiology and the pathophysiology of neuropsychiatric disorders. He has published over 400 scientific articles, over 180 reviews, chapters, letters, and editorials, and has received continuous NIH funding for 30 years, which is a remarkable accomplishment. We are so proud to have him here today to present his lecture, Schizophrenia, a Failure of Neuroplasticity. Dr. Coyle.
Well, first of all, I'd like to thank Cheryl Draper for the very kind introduction and the selection for this year's Ira Black Memorial Lectureship. This is a great honor, not only to walk in the steps of the previous speakers, Lloyd Green and Moses Chow, but especially because Ira was such a close friend of mine for 40 years. And I'd like to thank Janet for endowing this lecture and having Janet and Reed, Ira's son, here for the lecture. So, being a clinician, I have to give my financial disclosures. I own no stock in pharmaceutical companies, and my only consulting relationships have to do with drug discovery and not drug marketing. Let me just reflect on a few memories of Ira. Ira joined Julie Axelrod's lab back in the late 60s to his postdoc, and his postdoc terminated just as mine began, and he was going off to Cambridge with Janet to live in a thatched roof house, if I remember correctly, work with us Iverson. And he and I talked, and it was kind of like, what's the most important thing? And we both thought it was development. And Ira said he was going to study what makes neurons differentiate and develop their neurotransmitter characteristics, because nobody had looked at that. And I said, well, I'm going to try to discover how they develop in the brain, the catecholaminergic neurons. In a way, this demonstrated the difference between Ira and me, and I think of Isaiah Berlin, who talks about foxes and hedgehogs. And Ira, and neither are better. Ira was a real hedgehog. He would drill down on a problem, do experiments where there was only one possible answer. And I guess, as you'll see my research, I'm kind of looking around to see how things relate to each other, to see if we can develop insights. He demonstrated that the cholinergic input to the sympathetic ganglia was responsible for its differentiation. He demonstrated the role of nerve growth factor, which was known before these studies, but not known in terms of how it affected transmitter differentiation of neurons. He showed that the trophic factors were not just working in the periphery, that they also worked in the brain and controlled the development. I remember in 1990 when he became the chairman of the Department of Neuroscience and Cell Biology here, he and I talked because he was trained as a neurologist, and I was curious about how he made this career decision. And it was very clear he made the career decision because of his deep love for neuroscience. He founded Trophics Pharmaceuticals with the idea that in the long run, and I was always thinking in the long run, understanding how trophic factors work may help us treat neurodegenerative disorders. And he showed that BDNF actually was involved in synaptic plasticity and synaptic neurotransmission in the hippocampus. And then he did this astounding paper in 1990, and that is he infused stromal cells into fetal brains and showed that they differentiated in neurons. Now, this was astounding, and I have to tell you that I think about 95% of the neuroscientists at that time thought it was bogus, but that maybe those were microglia or they're astra, they can't really be neurons. But that shows you how visionary he is, because right now, that is the technology that's going to help us solve or help us provide answers to many of our disorders of the brain. And finally, I was invited down here as he became the director of the Stem Cell Institute in New Jersey. And anybody that knew Ira, he had a certain reserve about him, but he had a passion, and he was passionately committed to putting stem cells on the map as a research target and a hope for better treatments here in New Jersey. So I'm going to be talking about schizophrenia, and I hope to point out that at many points in our research on this issue, I keep on bumping into Ira, and how his discoveries have actually shaped much of our research. Now, let me tell you why you should care about schizophrenia. It's a disorder that affects. What happened? Okay, oh, great. It's a disorder that affects about 1% of the population. Its onset is early in adulthood, but it's a lifetime disability. It's the 7th most costly medical illness to society because almost all patients affected with schizophrenia remain out of the workforce. There's very high rates of substance abuse, homelessness, and imprisonment. And in Boston, the highest concentration of schizophrenics is not at McLean Hospital, but at the homeless shelter. And it's associated with premature mortality, not just because of suicide, but because of probably other systemic problems. Now, schizophrenia affects multiple domains, and it has what we call positive symptoms. Those are symptoms that ordinary people would not exhibit, such as delusions, hallucinations, thought disorder. These are very dramatic. These are what you see when you're walking on the street and someone may be shouting and gesticulating and they don't have a cell phone on. That's the type of behavior you see. Now, these symptoms tend to be relatively responsive to antipsychotic medications. But there are negative symptoms involving asociality, apathy, poverty of thought, anhedonia, emotional blunting. These symptoms persist through life, and they actually tend to get worse as time progresses, whereas the positive symptoms tend to diminish. And then there are positive deficits in memory, problem solving, executive function. Now, it's often kind of difficult to conceptualize what these negative symptoms really look like. And so I have a little bit of a tape I want to show of a patient being interviewed by Nancy Andreasen, I think the only psychiatrist to win the National Medal of Science. And let's just... So this patient has been treated with antipsychotics and has gotten pretty good response.
Brought you into the hospital when you first came here for this admission?
That's a bad question. I swear, I can't even remember.
So, simple question, can't come up with the answer. Very blunted affect, but as we'll see, he's not hallucinating.
A week or so ago, you mentioned that you felt as if, you were just on the verge of hearing voices again. But you don't feel like that now?
No, don't.
So lack of drive, disorganization.
One thing that you've mentioned that bothers you a lot is The fact that you can't seem to get yourself to do anything.
Yeah.
Can you describe what that's like at all?
I just can't do anything. I don't know why. I just can't seem to enjoy anything or do anything. And it seems to be getting worse.
The inability to experience pleasure.
Oh, going out on activities, volleyball, and you went to a movie today and so on. Did you enjoy any of those things?
No, I don't. Don't at all.
Social
You feel
isolation.
close to people or involved with them. Can you?
That's going away. I can't do that anymore. Diminishing.
And finally, cognitive impairment.
Can you name 5 things that start with the letter A?
Apple, ant.
So, is there any question why someone so affected would become estranged from their family, isolated from friends, unable to hold a job? And this is what about 75% to 80% of the patients are left with when they get the effective treatments or the relatively ineffective treatments that we have presently. So let me just, all right, so the dominant theory about schizophrenia is that it's due to overactivity of dopamine receptors because the drugs we use to treat schizophrenia, the antipsychotics, block dopamine receptors. There's a couple of problems with that theory. Number one, psychosis is not specific for schizophrenia. It occurs in bipolar disorder, depression, and we use the same drugs to treat that psychosis. Number two, it doesn't cure schizophrenia. I showed you what a patient who has responded to treatment looks like. And for 5 years, I ran the schizophrenia outpatient clinic at Hopkins, and I became very, how should I say, convinced that the dopamine hypothesis was missing its mark. So let's talk about what really occurs in the brains in schizophrenia. Now with postmortem studies and brain imaging, we know there's reduced cortical volume due to loss of gray matter, and this is particularly severe in frontal and temporal cortex. It's present at the onset of when the psychosis at first appears, and it gets worse over the next 10 years. There's a reduction in a marker called N-acetylaspartate, which is a marker of neuronal health, so the neurons are not healthy. There's reduced dendritic complexity and spine density. Here's a comparison of schizophrenia to normal in the cortical and hippocampal pyramidal neurons, which means there is reduced neuronal connectivity in the cortex. And these cortical changes correlate with the enduring symptoms of schizophrenia, that is, the negative symptoms and the cognitive symptoms, and they have no correlation with positive symptoms. So let's talk about glutamatergic neurotransmission in schizophrenia and not dopamine. This is a schematic representation of a glutamatergic synapse, and what's kind of neat about them is the menage a trois. It's just not a presynaptic and a postsynaptic, but the astrocytes also play an important role. And so glutamate is released, and it activates postsynaptic receptors. These receptors are either ligand-gated ion channels, cation channels, or GCPRs. And the glutamate-gated ion channels come in 2 types, the AMPA receptor and the NMDA receptor. Glutamate's action is terminated by being taken up into the astrocytes, not into the presynaptic neuron like norepinephrine. This NMDA receptor, as we'll see, requires another type of agonist in addition to glutamate, either glycine or D-serine, and their activities are in part regulated by the astrocyte. So the NMDA receptors, and what we're going to focus on, it really plays a very special role in the brain. At one level, it's what we call a coincidence detector. It requires several things to happen at the same time in order for it to do its job, and that is to allow sodium and calcium rush in. That tells me that it's a pretty important receptor because it has to be so tightly controlled. But at a more fundamental level, this is a receptor that regulates synaptic plasticity. And so this is a little cartoon, live cartoon. This is what postdocs are for in part, demonstrating how the NMDA receptor works. And so the NMDA receptor is silent when the neuronal membrane is not depolarized because the channel is blocked by magnesium. But when the neuron gets excited and depolarized, the magnesium block is eliminated, and so the receptor can be active and can gate cations. The receptor then responds to glutamate released at the synapse, but that still doesn't open the channel. There has to be a second agonist or coagonist, either D-serine or glycine, and when they are both bound there, then sodium and, more importantly, calcium rushes in, which drives gene expression, and primarily genes that encode for proteins related to synaptic function. And finally, a drug that will come up in the talk is ketamine. This is a dissociative anesthetic, and it serves as a non-competitive antagonist at NMDA receptors in the open channel conformation. So how did the NMDA receptor become implicated in schizophrenia? Well, this occurred almost 20 years ago. But John Crystal, well, I should say that parenthetically, if you worked in the emergency room in the 70s and early 80s, psych emergency room, we had an epidemic of young people coming in looking like they had schizophrenia, but they had been smoking ketamine, super K, or taking PCP, phencyclidine. And we knew those were dissociative anesthetics, but it was unclear whether they were looking like they had schizophrenia because that's who would take these drugs, and they really had it, or the drugs themselves were causing it. Then John Crystal did a study at Yale in which he took normal volunteers, they're Yale undergraduates, not quite up to the tier of Rutgers undergraduates, I'm sure, and infused them with a constant low dose of ketamine. And what he found was this replicated the positive, negative, and cognitive symptoms seen in schizophrenia. Then subsequent studies used this technique and showed that this blocking of NMDA receptors replicated the physiologic signs of schizophrenia, an inability to activate the frontal cortex when cognitive challenges, elevated subcortical dopamine release, I said dopamine again, and eye tracking abnormalities and disrupted gamma rhythms. We were working on this dipeptide, N-acetylspartyl glutamate, and found which blocks NMDA receptors and activates receptor mGluR3, which inhibits glutamate release, and found that it was elevated in postmortem brain and the enzyme that degraded it was reduced in schizophrenia. And my first postdoc, Robbie Schwartz, has focused his career on metabolites of tryptophan that interact with NMDA receptors, and kynuritic acid is an endogenous metabolite of tryptophan that blocks NMDA receptors, and it's elevated in schizophrenia. And then just recently, there's been this syndrome described, which is an autoimmune disorder in which the individuals make antibodies against the NMDA receptor. And in many cases, these individuals present with a schizophrenia-like syndrome. It's very eloquently described in this book that was published last year, Brain on Fire, by a New York Post reporter who developed this syndrome but was ultimately cured. So that's sort of last century stuff, except the NMDA receptor antibody findings. drug challenges post-mortem. Now, as we get post-human genome, it's clear that schizophrenia is is fairly highly heritable, and there's been very serious searches for what the risk genes might be. Now, what has become clear in these studies is that it does not follow Mendelian genetics, that this is a disorder of complex genetics, as most psychiatric disorders are, where multiple genes of modest effect interact to result in the phenotype. And there's this site, SchizGene, which keeps a running meta-analysis. And here are some of the genes that are within 2 degrees of separation of the NMDA receptor that are risk genes. D-amino acid oxidase is the enzyme that degrades D-serine, and it has a separate modulator. They're both risk genes. Serine racemase, which makes D-serine, is another risk gene. And there are several other of these genes, and I'll show you how they interact with NMDA receptors, that are risk genes for schizophrenia. So we can see that the genetic findings are corroborating the clinical and postmortem findings. And then just last year, Kirov and colleagues looked at subjects that have a type of mutation called a copy number variant, where there is either a deletion or a duplication of a significant portion of DNA. It typically involves several genes. And these are associated with older fathers and may account for the fact that schizophrenia doesn't disappear in spite of the lower fertility, because there may be new mutations constantly being introduced. But anyway, they looked at the genes that were associated with the CNVs, and the CNVs were enriched for members of the NMDA receptor complex. Our data indicate that defects in NMDA receptor postsynaptic signaling, which are known to be important in synaptic plasticity and cognition, play a significant role in the pathogenesis of schizophrenia. So let me just schematically insert these risk genes. And so what we're talking about here, again, are not Mendelian mutations. We're talking about allelic variants that would have modest effect, but if you had several of these in a row, it could have, one would presume, substantial effect. And so here is, let's see, yeah, okay. Serine racemase, which makes D-serine a coagonist at MDA receptor. G72, which modulates D-amino acid oxidase, which degrades D-serine. D-amino acid oxidase itself. The NR2B, oops. What the heck? Oh. The NR2B component of the NMDA receptor. Neuregulin, which has multiple actions, but one of them is interacting with the NMDA receptor. Despindin, which is concentrated presynaptically in glutamatergic neurons, regulates glutamate release. And what has now popped up in the GWAS studies is mGluR3, which downregulates glutamate release. It's one of these metabotropic glutamate receptors. So again, you can see this is a pathway that would interfere with NMDA receptor neurotransmission. I just want to emphasize that D-serine is concentrated in the cortical limbic regions of the brain. This is the rat brain, but it's the same thing in humans. And these regions have rather low levels of D-amino acid oxidase activity, where it's very high in cerebellum brainstem, and there's very little D-serine there, and glycine is the likely co-transmitter. So in trying to understand what alterations in NMDA receptor function might mean, we created a mouse in which we knocked out serine racemase. And our rationale was that there are 3 of these genes that directly regulate the availability of D-serine. It's been shown that D-serine is reduced in the CSF and plasma in schizophrenia. Postmortem studies indicate that DAO is elevated, And as I showed you, D-serine and serine racemase itself are concentrated in the cortical limbic regions of the brain, the very regions that are affected by this atrophy in schizophrenia. So this is just shows you classic knockout. Here's serine racemase staining in the brain. It's a cortex limbic system. Here's the knockout. And this results in a greater than 85% decrement in D-serine in the brain. And using a biochemical study that summates NMDA receptor by looking at nitrosylation, we see about a 70% reduction in NMDA receptor function in the cortex. And let me emphasize that there's still glycine in the cortex, so you have a system that's impaired, but it's not entirely broken like a mouse in which the NMDA receptor was knocked out that Caron made about a decade ago. So we're going to use, in part, electrophysiologic techniques to look at glutamatergic synaptic neurotransmission. And so you take an acute slice, and you can record from the pyramidal neuron, and you can stimulate known glutamatergic inputs to the pyramidal neuron. You can perfuse with drugs so you can isolate individual receptor currents, like the NMDA receptor current or the AMPA receptor current. And so what happens in these knockouts? Well, we looked at at the current NMDA currents at the CA1 pyramidal neurons when you perfuse with D-serine. And in the wild type, you get a little bit of boost in an NMDA current, but in the knockouts, you get this very robust boost, indicating that this glycine modulatory site where D-serine acts is not fully saturated. And you can also see it with a drug called NFPS, and I'll tell you a little bit more about that in a second. In addition, there's this form of functional synaptic plasticity called long-term potentiation. And this occurs at glutamatergic synapses. It's mediated by the NMDA receptor. And the way it's shown is if you stimulate the glutamatergic input at a modest rate, stimulate presynaptically, you get bing, bing, bing. You record postsynaptically, you get a faithful representation, bing, bing, bing. But if you stimulate it really fast, really hard, called tetanic stimulation, before you do it, bing, 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 bing. After you do it, bing, bing, bing. Postsynaptically is boom, boom, boom. So there's a tremendous increase in excitatory neurotransmission at the synapse. And this is associated with memory formation and is thought to be sort of a molecular physiologic a manifestation of a memory. And so in the knockouts, in the wild type, you get this, when you do the stimulus, you get this big postsynaptic response after the robust stimulus. In the knockouts, it goes down about 75%. If you perfuse a slice with D-serine, you can restore it back to normal. And when we looked at the cognition in these mice, they could learn a memory task called water maze, like the wild-type mice. But when you moved the platform, they were unable to very efficiently sort out what was going on. So you have to remember that schizophrenia is not mental retardation. These individuals have IQs that are about 5 to 10 points below what you would expect because of their SIBs. But, they can navigate pretty well. And so this subtle type of cognitive impairment is consistent with the phenotype. And we're interested in the relative contribution of D-serine versus glycine to NMDA neurotransmission in the cortical regions where D-serine appears to be so prominent. In a study with Yang Li that was just recently published, we found that D-serine seemed to be responsible for relatively low levels of activity, but when you looked at the knockout glycine would be recruited when there was high levels of presynaptic activity. So glycine still is able to work, but it is sort of working under high demand conditions. So for those who are in the cognoscenti about glutamatergic neurotransmission, one of the widespread beliefs from various studies is that D-serine is synthesized and released from astrocytes. Actually, Roger Nicole wrote an editorial about 6 months ago in Cell, starting out. As everyone knows, D-serine is synthesized and released from astrocytes. But knowing where D-serine comes from is important if you have a disorder of D-serine deficiency, how you might, in fact, deal with that. And so we looked at conditionally knocking out serine racemase in astrocytes versus the glutamatergic neurons in the forebrain using Cree, which is the thing that clips the gene out, driven either by an astrocyte promoter or by a promoter expressed exclusively in the pyramidal neurons. And so I would send the postdoc off to do the experiment, and comes back to lab meeting next week and says, I don't see anything when I look at the astrocyte neurons. So I figured this is you go after the low-hanging fruit. Do it again. You're not using enough this. You're not doing enough that. And after we had about 25 animals, we could show that there was about maybe a little bit over a 10% decrement in serine racemase. But when we knocked it out in the glutamatergic neuron, there was a 65% reduction in serine racemase. So there's virtually no serine racemase in the normal astrocytes in the adult brain, and there's a lot in the pyramidal neurons. And this just shows you a human brain. These are pyramidal neurons. Here's CAM kinase, which is the marker for them, and their serine racemase is co-localized in the pyramidal neurons. We also looked at D-serine. Actually, these are this is a marker for neurons. Actually, this is a GABAergic interneuron, and you can see they're co-localized. Here's D-serine and serine racemase. And what we found, interestingly, is that D-serine is not always co-localized with serine racemase. It can be in about a 50% overlap, and the serine racemase cells are a greater number than the D-serine positive cells. So bottom line is that serine racemase in the normal adult brain is expressed in the GABAergic interneurons and in the pyramidal neurons. Both neuronal systems express the transporter that concentrates D-serine, and astrocytes do not. So what happens in these animals? We found that their cortical volume was reduced by about 3.5%. And that's about precisely what you see in schizophrenia. Again, it's not mental retardation. It's not profound cortical atrophy like you see in Alzheimer's disease. But I don't think you'd want to give up 3% of your cortical volume. And then we looked at the in Golgi stains, something I never expected. I'd be using a technique that was discovered 120 years ago, but it is a very powerful technique. And what we see, and this is in primary sensory cortex, is a significant reduction in dendritic complexity, which means they have shorter dendrites, and a significant reduction in spine number, which are the synapses. And if you multiply density times length, what this indicates is that in the schizophrenic, there's probably between a 30 and a 40% reduction in synaptic connectivity of the pyramidal neurons. This is from a study that just came out in PNAS, where we looked at the hippocampus. I should also say we looked at prefrontal cortex, seeing similarly reduction in dendritic complexity and spine number. This is looking at the granule cells in hippocampus. We can't look at dendritic complexity, but we can look at spine density, which is reduced. And Here I am bumping into IRA because the BDNF RNA is reduced, BDNF is reduced, while track B protein is not reduced, phosphotrack B is reduced, and a microRNA that regulates synaptogenesis, microRNA 132, is also reduced. And I should point out that all these findings are what's found in schizophrenia. So if you look at the postmortem studies in schizophrenia, cortical, and compare to serine racemase knockout, cortical atrophy, reduced dendritic complexity, reduced spines, reduced BDNF, phosphotrack B, phospho-AKT, phospho-CREB, reduced miR-132, and subtle cognitive impairments. Now, as you are all aware, when you do a constitutive knockout, the animal has not seen the gene product since conception throughout development. And so we were interested in whether it had to be throughout development, or if you just shut down serine racemase in adulthood, would you see changes in dendritic spine complexity and in spine density. And so we used these conditionally expressing CAM kinase driven Cree to turn off serine racemase in the pyramidal neurons. And while the phenotype is not as robust, you still see significant decrease in synaptic complexity in the basilar dendrites and a significant decrease in spine density in the apical dendrites. So these are animals in which they had normal serine racemase expression until about 5 weeks after birth. Now, there's one other pathology that is unequivocally present in schizophrenia, and it has to do with the downregulation of a subpopulation of GABAergic interneurons. These are the parvalbumin, fast-firing GABAergic interneurons. They regulate the output of the pyramidal neurons. They get recurrent innervation from the pyramidal neurons, and it's become quite clear that they coordinate excitatory neuronal activity in the cortex and regulate gamma rhythms, theta rhythms, etc. When you look at postmortem studies, the enzyme 4 making GABA in the intermediate layers is reduced. If you look at parvalbumin, it's reduced, but a marker for these more superficial GABAergic interneurons are not reduced. So what does that have to do with NMDA receptor? Well, John Listman, who's part of our Conti Center, an electrophysiologist, subchronically treated rats with ketamine and looked at what happens in their prefrontal cortex, and lo and behold, you see the downregulation of GAD67 and parvalbumin, like you see in schizophrenia, and he showed that there was reduced inhibitory synaptic currents. So this downregulation would dysregulate pyramidal neuronal firing. So the last issue I'd like to touch on is, can enhancing NMDA receptor function alter the symptoms of schizophrenia? Now, no drug companies were making drugs to activate NMDA receptors because, in fact, that was the third rail. Because if you overstimulate NMDA receptors, neurons die. It's called exocytosis. And so there wasn't much out there, but we're opportunistic. And there was this drug, D-cycloserine, which was used to treat tuberculosis. It crosses the blood-brain barrier. It's a partial agonist at this glycine modulatory site with 50% of the efficacy. Downside at high concentration, it inhibits serine racemase, and it's been shown in experimental animals to have neuropsychological effects. So this is a study done with Don Goff at MGH. It's a blinded dose-finding study with D-cycloserine, looking at two things, negative symptoms and a cognitive task. And what you see is at 50 milligrams per day, there was a significant improvement in negative symptoms and a significant improvement in performance on the cognitive task. Don recounts that one of his patients, he knew all these patients extremely, one who had never done anything with his family showed up one day and said, I can't come to clinic next week because I'm going on vacation with my family. So this was very striking, and there have been a number of studies that have confirmed that, but also have not confirmed it when it's used long-term because you get desensitization with D-cycloserine. Now here I bump into Ira again. So when we published these findings, a lot of skepticism because the concentration of glycine in the spinal fluid was way above the amount that would saturate the glycine modulatory site. So an agonist should not have any effect. The site should be saturated, and especially a partial agonist should make it worse. And so one of the agents that Trophics developed was NFPS. Talk about vision. This is back in it's probably 1996. So it was known that sarcosine inhibits the transporter for glycine. And so what was done was take sarcosine and drugify it, fluorine, benzenes, and it gets in the brain and is a very, very potent inhibitor of the glycine transport. And so when you look at NMDA neurotransmission at that CA1 neuron, this is a baseline. This is the activity you get. If you give very low concentrations of the glycine agonist, you get an increased NMDA occurrence. And if you give glycine and NFPS, you get even more robust increase in currents. Well, that told us two things. First of all, it told us that the glycine modulatory site was not saturated, that the transporter was able to maintain subsaturating concentrations of glycine at the synapse. And secondly, it told us that this glycine modulatory site was a potential target for treatment. We also looked at D-serine, and this is, I should point out in these studies, we added on to stable antipsychotics and showed a reduction overall symptoms, reduction in negative symptoms, improvement in cognition, and even some improvement in positive symptoms. So what is happening with the drug? We did functional imaging to see how the drug was affecting cognition. And so this was a memory task that was performed on, off, or performing, not performing. And W. Ergelin Todd did fMRI on these subjects. And when they performed the task, there's an activation of the left superior temporal gyrus. Here it is right here. Now, when schizophrenics are not receiving D-cycloserine, they don't get the activation. When they're on it, they do get the activation like wild type, or I guess normals, we're humans. And so there's this robust activation on the D-cycloserine, and this activation correlates with the reduction in negative symptoms. And so one can see that enhancing NMDA receptor function enhances the performance by activating the site in the brain required for the performance. And so Bo-Chen Tsai, who got his PhD with me and did a postdoc with me, trained as a child psychiatrist, recently published on a meta-analysis of placebo-controlled glycine modulatory site agonist clinical trials in schizophrenia. So about 800 subjects from 26 studies were included in current meta-analysis. Overall, the NMDA enhancing molecules are effective in most schizophrenic symptom domains with effect size and total psychopathology of 0.4, and an effect size on depression, negative symptoms, cognition, positive symptoms, and general psychopathology. The results demonstrate highly significant effects by giving agents that enhance NMDA receptor function. So I mentioned the reduction in BDNF, and BDNF acts through the TrkB receptor, which when activated gets phosphorylated, and then it can activate these downstream pathways. And we have found that the pathway that involves phosphorylation of AKT, phosphorylation of mTOR, and ultimately phosphorylation of Krebs appears to be involved in the response. And in addition, recent studies indicate that voltage-sensitive calcium channels are involved not only in interacting with TrkB, but also in promoting the synthesis release of BDNF. And quite interestingly, there are two voltage-dependent calcium channels that on the Manhattan plots look like the Twin Towers in GWAS studies. So we want to know what happens if we normalize D-serine. We treated them chronically with D-serine in the periphery, 1 injection per day, and you could normalize in the hippocampus and nearly normalize it in the cortex in the serine racemase knockout mice. And what we were able to see is that when we normalized D-serine, we could normalize the BDNF, phospho-AKT, we could normalize phospho-mTOR, and the phospho-GSK, GS3KA alpha and beta. So these markers for BDNF hypofunction can be corrected by presynaptically, by restoring D-serine levels. In the serine racemase knockout mice, I showed you have low LTP. If you treat the animals, and this is just taking the animal that's been treated, removing his hippocampus, not giving him an exogenous D-serine in the bath, and you restore LTP. And finally, we can see that restoring D-serine levels restores the cognitive impairments in memory deficit, hippocampal-associated memory deficits in both cue and context-dependent performance. The last point I want to make with regard to this is D-serine is not a great drug. You'd have to eat a lot of D-serine every day. It also has potential toxic effects on the kidney. So we're interested if we could work around the NMDA receptor and try to correct these deficits. And Jeff Kahn has made a mGluR5 positive allosteric modulator. mGluR5 promotes the phosphorylation of the NMDA receptor and makes it more responsive to normal input. And this shows that, like D-serine, we can restore the deficits in the downstream markers to BDNF that occur in the serine racemase knockout mouse. And so, you know, I focused on the NMDA receptor, but in fact, schizophrenia is not a disorder simply of a receptor. It's a circuit-based disorder. And I've given you many examples of ways that NMDA receptor hypofunction can be induced. Ketamine, the endogenous inhibitors, NAG or kynurenic acid, or mutations in a number of genes that directly affect the availability of D-serine or NMDA receptor function. And these lead to NMDA receptor function and can account for the dendritic dysplasia, as well as the disinhibition of glutamatergic output because of the reduced activity of GABAergic interneurons. And this will drive subcortical dopamine release, and this has been shown in imaging studies, resulting in psychosis. Meaning that psychosis, which has been the focus for the last 50 years, is a downstream event from primary cortical pathology. And the last point I want to make, again, bumping into Ira, was our recent work with Sang-Mi Chung, in which she has been able to take human inducible pluripotent stem cells, and through manipulation of trophic factors cause them to differentiate into median ganglionic eminence, parvalbumin positive, GABAergic interneurones. And what Ira saw 13, 14 years ago is now becoming fairly routine in laboratories, and I think this will replace, to a large extent, our mice with methods to actually look at individual pathophysiology of schizophrenia. So pharmacologic postmortem genetic results indicate that hypofunction of NMDA receptors is central to the pathophysiology of schizophrenia. NMDA receptor hypofunction impairs neuroplasticity, causing spine loss and dendritic atrophy. We think due primarily or significantly to BDNF deficits. Acute pharmacologic enhancement of NMDA receptor function improves negative symptoms and cognitive impairments, but we believe this is the holy grail. Long-term enhancement of NMDA receptor function may restore markers of plasticity and cognition in schizophrenic individuals, and as it does in our serine racemase knockout mice. And I just want to recognize the people who have really done the work, my collaborators, current postdocs, and of course grant support. And finally, again, I want to thank Janet and Cheryl for having me here to continue to remind you of the very special person that Ira was, very special scientist, and keep that memory alive at Rutgers. Thank you.